Hello to everyone interested in the fascinating ecology of periwinkles and rocky shore habitats. My name is Grace Weston, a fourth-year marine biology student at Dalhousie University, and today I'll be presenting a study analyzing periwinkle shell size across rocky intertidal habitats at Lawrencetown Beach in Nova Scotia. This study took place at Lawrencetown Beach Provincial Park, located on the eastern shore of Nova Scotia. It is an exposed Atlantic coastline characterized by a mixed sand and cobble intertidal ecosystem. This type of habitat is well suited for intertidal organisms, including periwinkles found in our study, due to the presence of rocky surfaces and tidal variation. Since the study was non-invasive and did not involve organism collection or disturbance below the high tide mark, no research permit was required and all provincial park guidelines were followed. The common periwinkle, Littorina littorea, that is being measured in this study is a small marine gastropod found in intertidal zones along rocky and mixed shorelines. It is native to the North Atlantic and is widely distributed across Europe and eastern North America. Periwinkles can be found from the upper intertidal zone down into shallow subtidal zones of around 40 meters deep. They are grazers on algae and other organic matter, and they play an important ecological role in coastal food webs. One of their key adaptations is their ability to tolerate severe natural conditions such as temperature and salinity changes and wave exposure, which allows them to survive in harsh coastal environments. I outline some of these environmental conditions later on in the presentation. This research was conducted in Mi'kma'ki, which is the unceded and ancestral territory of the Mi'kmaq people. The Mi'kmaq have been and continue to be the caretakers of these lands and waters for thousands of years. This acknowledgement recognizes their long-standing relationship with coastal ecosystems and their ongoing stewardship of the environment where the study took place. The main question the study proposes was whether periwinkles occupying different tidal zones differ in size. Therefore, the null hypothesis states that there's no significant difference in maximum shell diameter of Littorina littorea among the high, mid, and low intertidal rocky shoreline areas at Lawrencetown Beach, Nova Scotia. Before fieldwork, data sheets for each transect were prepared in a field notebook, and a low tide time period was selected for Lawrencetown Beach to maximize access to the chosen sampling area. I also constructed 2.5 by 0.5 meter quadrats by cutting PVC pipes and assembling them with elbow connectors for data collection. Periwinkle sampling was conducted within the rocky intertidal zone at Lawrencetown Beach on May 30th and May 31st, 2026. 3 transects, each 8 meters long and spaced 15 meters apart, were established across the high, mid, and low tidal zones using a 30-meter measuring tape, small garden stakes, and flagging tape to mark transect distances as 0, 4, and 8 meters respectively. A total of 27 quadrats were sampled, with 3 quadrats placed within each tidal zone along every transect. Quadrats were placed around 2 meters apart for every transect. Important field equipment included in the study was the custom-made PVC quadrats, a caliper for shell measurements, a GPS app used to record locations, a waterproof digital thermometer, and a salinity refractor for environmental data. To reduce sampling bias, each quadrat was divided into 4 equal 0.25 by 0.25 meter subquadrats labeled A through D, and a random letter generator was used to select the area that was going to be sampled. Within the selected subquadrat, periwinkle sizes were measured using a caliper. Shell size was recorded as the maximum shell diameter, measured as the distance between opposite sides of the widest part of the body whorl in millimeters. Additional observations included tidal zone, substrate type, abundance, and GPS location. Environmental measurements, including temperature and salinity, were collected from each tidal pool zone along all 3 transects using the digital thermometer and salinity refractor for a total of 9 environmental samples each. Following data collection, shell sizes were grouped by tidal zone and analyzed in RStudio. A one-way ANOVA was used to test for differences in shell size among rocky tidal zones after checking assumptions of normality and homogeneity of variance. Because this was an observational field study, there was no permit required to access the site. Additionally, there was no controlled treatment. Instead, naturally occurring high, mid, and low tidal zones were compared instead. Following the data collection, the results show a total of 52 periwinkles were measured for size across the 3 tidal zones. Figures 2 and 3 show the mean shell diameter for each tidal zone, as well as error bars representing variation within each group. The low tidal zone had the largest average shell size at 8.82 plus or minus 3.96 millimeters, followed by the high tidal zone at 7.57 plus or minus 2.91 millimeters, and then the mid-tidal zone had the smallest average shell size at 6.89 plus or minus 2.39 millimeters. The plots show a decrease in mean shell size from the high zone to the mid-zone, followed by a slight increase in the low zone, and there was substantial variation among individual periwinkles within each group as shown by the overlapping error bars. The largest individual measured during the study was 16.2 millimeters, represented in Figure 2, found in Quadrat 8 within the low tidal zone. Several other large individuals were also found in the low zone, including shells measuring 11.5, 10.2, 10.1, and 9.9 millimeters. Opposite trends were found higher on the shore, with some of the smallest individuals measuring 2.5 millimeters in Quadrat 10 and 3.1 millimeters in Quadrats 3 and 23. For statistical analysis, a one-way ANOVA was performed to compare shell size among high, mid, and low tidal areas of the rocky intertidal shoreline. The analysis produced an F-value of 1.78 and a p-value of 0.18. Assumptions of the ANOVA were tested before interpreting the results. Homogeneity of variance was evaluated using a Levene's test and RStudio, which produced a p-value of 0.646. Figure 4 shows a residuals versus fitted values plot with a relatively linear horizontal pattern. Normality of residuals were also assessed using a Shapiro-Wilk test, which produced a p-value of 0.415. A QQ plot in Figure 5 also showed the residuals closely following the reference line. All p-values exceeded the significance value of 0.05, and therefore the ANOVA results were considered valid for interpretation. Environmental measurements were also collected from each tidal zone along all 3 transects. Temperature measurements range from 10.1 to 11 degrees Celsius in the high tidal zone, 9.6 to 10.4 degrees Celsius in the mid-tidal zone, and 8.5 to 9.8 degrees Celsius in the low tidal zone. Salinity measurements ranged from 26.9 to 32 parts per thousand in the high tidal zone, 32.5 to 35 parts per thousand in the mid-tidal zone, and 35 to 35.2 parts per thousand in the low tidal zone. Additional abundance observations were recorded during sampling. The highest abundance was observed in Quadrat 13, where 43 periwinkles were counted. Several low zone quadrats, including Quadrats 9, 17, and 18, contained no periwinkle data at the time of sampling. Although tidal zone was not found to have a statistically significant effect on shell size, several biological patterns were observed that help explain the trends shown in Figure 2 and 3. The low tidal zone contained the largest average shell size and had the largest individual measurement during the study at 16.2 millimeters. The smallest individuals, measuring between 2.5 and 3.1 millimeters, occurred at higher locations. Figure 3 illustrates this overall increase in shell size towards the low tidal zone, although the overlapping error bars indicate considerable variation among individuals. The ANOVA result, with an F-value of 1.78 and a p-value of 0.18, indicates that these differences were not statistically significant. However, the pattern observed may still be biologically meaningful and could become clearer with a larger sample size. Environmental sampling suggests a decrease in temperature along all transects moving down to the shore, while salinity increased. For example, temperatures measured around 11 degrees Celsius in the upper tidal areas to 8.5 degrees Celsius in the low tidal zone while salinity increased from a minimum of 26.9 parts per thousand to a maximum of 35.2 parts per thousand. These colder, more saline conditions may favor the growth and survival of larger individuals. Abundance patterns also showed an interesting contrast between quadrats. The most abundant sample was quadrat 13 in the mid-tidal zone, where approximately 43 periwinkles were recorded. Meanwhile, several low-zone quadrats contained little to no individuals. Despite these lower abundances, the largest shell size were consistently found in the low tidal zone. This suggests the area supporting fewer periwinkles may contain larger size classes, likely due to reduced interspecific competition, differences in habitat condition, or different exposure levels. These observations are consistent with previous studies, such as Ellis 2007, who found periwinkle abundance often reaches its highest level in upper intertidal habitats. Similarly, I observed greater abundance in upper and midshore quadrats compared to many lower shore locations. Etchweiler 2009 reported that larger periwinkles are often associated with rocky habitats and that larger size classes are more common in lower intertidal zones, while higher density populations frequently contain smaller individuals. Their study also found that abundance on sedimentary shores was approximately 3 times higher than on rocky shores, while average shell size was considerably smaller. This closely aligns with trends observed in my study, where larger individuals tended to occur in lower density areas. Hilberg and Christensen 1978 also found that average periwinkle size increased with environmental exposure. They suggested that larger individuals may be more tolerable to physical stress because of their strong attachment ability and reduced competition from smaller gastropods. Since Lawrencetown Beach is an exposed Atlantic shoreline, these mechanisms may partially explain why larger individuals were associated with lower intertidal habitats. This study had several limitations. Sampling was restricted to low tide periods and a short time frame, which limited replication and reduced coverage of quadrats where no periwinkles were present. In addition, environmental measurements were collected on a separate day than the biological sampling under colder, wetter, and windier conditions, so temperature and salinity values may not fully reflect the conditions experienced by the measured periwinkles. Future research should increase sampling across multiple tidal cycles, seasons, and shoreline zones to determine whether these patterns persist over time. A larger dataset would also allow analysis of how shell size relates to environmental factors, such as temperature, salinity, wave exposure, and abundance. Incorporating a two-eyed seeing approach by combining Western scientific methods with Mi'kmaq knowledge could further strengthen our understanding of intertidal ecosystems and support collaborative coastal stewardship within Mi'kma'ki. During my fieldwork, I also made observation of many other organisms that were not relevant to my study whatsoever, but I thought they were really cool and that I'd add them into my slides. There is one photo that shows coralline algae growing on the largest periwinkle measured in the study. This demonstrates how shells can act as a substrate for other organisms and how organisms can benefit from one another in rocky intertidal habitats. To conclude, the results of the one-way ANOVA showed no statistically significant difference in periwinkle shell size among the high, mid, and low intertidal zones at Lawrencetown Beach. The analysis produced an F-value of 1.78 and a p-value of 0.18, which was greater than the significance level of 0.05. Because the p-value exceeded 0.05, I failed to reject the null hypothesis, meaning there was insufficient statistical evidence to support that different tidal zones influenced the maximum shell diameter of Littorina littorea within the study area.